Great. So I think everyone's online and we're here, so we're recording, right?
Yep.
Yep, great. Okay, so welcome back everyone for the second class or the first lab from the single molecule sequencing course. And in addition to what Theo's been working on and you all heard about last week, hello, come over. We're just getting started. Come on in. We're now going to do some of the labs. Actually, this is one of my favorite things. As I spent many long nights staring at BAM and SAM files and formats, and so this is one of the things that once you learn once, you will always use as long as there's sequencing, which there will always be sequences because most of life is based off nucleic acids that we know of. So until someday when we find and or make life in different planets that is made of different molecules, then you'll have to take a different class. Until then, this will be the formats and data structure you'll use for many projects throughout probably the rest of your life. And in particular, I want to introduce Karolina Sienkiewicz, hopefully I'm pronouncing it. Sienkiewicz, who is a wonderful grad student, not only in my lab, but also in Ari Milnick's lab, and is a PhD candidate. But also, I think really importantly, she has been for several years doing an NGS school and teaching people how to use some of the same tools, methods, computational algorithms. And will be joining us as well to help get the course and help get people familiar with some of these tools with Theo and myself, and also as a resource, not only for our lab, but also for the class. And I think did you another slide on the intro?
Yeah, I put here the icon on the right. It's the or left, depending on how you look at it. Yeah, it's SLOW5. It's my program for multi-omic data integration. I will tell you more about it in like incoming module. And we have a schedule on the next slide. So I put your class project introduction here in week 5, but basically, since I am coming from the world of not single molecule sequencing, but from like integration and like feature level integration and a lot of coding, I think my most important role here is to support you in terms like technical.
Welcome back. So I just sent out a notebook on Google Colab. If you're looking at this on YouTube later, it'll also be available in the description. That should be sent to the whole class so everybody can take a look. The first step is mounting your Google Drive. You should have been able to go through that during homework 1. Did anybody have difficulty going through that with homework 1? Great. So it's the same setup, and then it goes through 5 different common formats. I'm going to do a little bit of a chalk talk just to introduce these formats and to try and situate them relative to one another. It's organized in the same way that a typical analysis pipeline would be in terms of all of the various steps, but it prioritizes knowledge about these specific file formats over necessarily the tools and the processes by which you convert from one to the other. We'll have plenty of time in the subsequent lectures to go through a lot of those tools. So the first sort of overarching program is this one called BioConda. And basically BioConda, how many people are familiar with it? Did I get a hand raise? Okay, pretty good familiarity. It's the repository of all knowledge, repository of all tools related to bioinformatics. And you probably are also familiar with, if you've done any next generation sequencing analysis, FASTQ and SAM-BAM. So BAM being the binary version of SAM in a sense that it allows for data storage and compression without loss of information. These are your standard file formats that you get from any next generation sequencing experiment. And there's reviews in here about how those are organized. In essence, on a summary level, they're row-based systems where each row corresponds to a different read or piece of information, or in the case of SAM-BAM, an alignment to a reference genome. The formats that are likely going to be newer are the FAST5, POD5, and SLOW5 / BLOW5 formats, with BLOW5 again being the binary version of the SLOW5 format. These are basically all various interchangeable formats that allow you and Nanopore to organize the raw signal information that comes through the Nanopore. So as you saw in the last slideshow, we had videos and lectures of individual nanopores, and we saw reads runs of either DNA or RNA moving through those pores. These file formats, in essence, need to, one, capture the signal information. Two, need to capture information about the pore. And three, need to capture info about the flow cell system. So on these various levels, the actual signal refers to what is going on inside of the pore as the individual molecule goes through. The pore itself has its own properties dependent on where it is, its own spatial location, how well it's functioning, how fast the motor protein on that individual pore is moving on average, et cetera, et cetera. And then the flow cell provides a whole bunch of overarching information about the system that you're running this particular sample on in this particular moment. And Nanopore makes updates to the parameters and configurations of their flow cells consistently. So it's good to have this information for when you're potentially aggregating meta-analyses across samples.
And we'll talk about next week, but RNA sequencing is slower, like 120 bases per second versus 450, which adds a big factor in how fast it goes. I'll make sure to talk about that.
And that's something that, yeah, is captured within these formats. As I mentioned, these are all analogous to each other, but FAST5 and POD5 are maintained by Oxford Nanopore. So these are the sort of company official sponsored formats, and then SLOW5, BLOW5, those are formats that were designed by a research group. So they're open source and they're community formats. They're organized in different ways. So as you'll see, FAST5 is organized into the HDF5 structure. How many people are familiar with JSON files? Have seen a JSON file? Not as much. So HDF5 can be thought of as analogous to JSON, where in essence, your information is organized into specific properties. So instead of having, an entire, like a row or a column that has all the information that you're interested in, you have this format that looks like a dictionary in Python, or that basically where you have keys which contain each individual attribute that you're interested in about a specific run, and then values that represent the data specific to the read of interest. And typically, the way that the HDF5s slash JSON adjacent files are broken down is on a per read basis. And so if you go back 2017, 2018, Nanopore was actually outputting sequencing information in FAST5 files that each contained a single read. So they would, if you had 1,000 reads coming out of a sample, you had 1,000 FAST5 files. Now the FAST5 format that they use is analogous to multi-line FAST5. So one FAST5 file can contain all of the reads, but organized pretty much in the same manner. POD5, by contrast, is their newer format because now they have this 004 chemistry that we'll talk about, another chemistry that generates tens of millions of reads. And POD5 is basically a column-based format for analyzing this data. So if you are thinking about it, you have columns. Each column is its own read. So read 1, read 2, read 3. And then all the information about the read is contained in this sort of column-like structure. And then you can compare across columns. So you also have sort of signal level information that goes down. And then the last format, the community-based format, why it exists, is that parsing data through these POD5 files or through the FAST5 files, it's pretty unintuitive for most bioinformaticians. And it's not aligned with, as I mentioned, the row-based systems that FASTQ and SAM implement. So since, that's how people are used to it, SLOW5 was basically developed along with a toolkit called FLOW5 tools that you'll use today in order to take this data and organize it more intuitively. So I will go through. So the first, as you saw in homework 1, there was an example SLOW5 file that had a single read in it. And hopefully, looking at that, you got a little bit of an intuition for how it was organized. The first 59, 60 lines are just metadata. So they contain all of metadata, they contain all of the information related to the specific flow cell of interest. And then each line is basically the information about the read. So you can see that the first column, for example, gives you information about the read identifier. So that's a unique ID assigned by the actual sequencer. And at the 8th column includes a list organized in a comma-separated format value to a CSV list of raw signal values that were output by the sequencer. Now, the raw sequencer values, even though those are the raw ones, are actually not the same as the electrical current signal. And there is a conversion rate that you need to do based on the properties of the pore. But all of that data is maintained. So one of your homework assignments will be to convert these raw signal values from the 8th column into their corresponding electrical current values, which then allows for signal comparison between pores, either on the same nanopore or between pores from other flow cells. So those are basically, on a high level, the different formats, how they fit together. And typically we have tools such as Bioconda to install all of these package programs. We, in a classical analysis pipeline, take the FAST5, BLOW5, SLOW5, BLOW5 signals, convert them to FASTQ, and then we use an alignment program to convert the FASTQ to SAM-BAM. Typically, the pipeline will keep going if we're interested in epitranscriptomics with a program called F5C, which allows basically for a connection between these alignments and their raw signals. So that's the role of F5C. And M6A is a program that, for example, can take the alignments and their interpretations of raw signal, travel this pipeline all the way to the raw signals, and use the raw signal to generate predictions. And these predictions being, is that specific base M6A methylated? You can do the same thing, for example, also with M5C methylations in DNA. And theoretically, any base pair modification can undergo this pipeline if you understand sort of what that specific base pair or how it's supposed to canonically alter the raw signal that exists. So this is the high-level objectives. Now, if you go through, we're gonna break out into a workshop style session where everybody takes the time to go through the notebook at their own pace. You'll see that at different points, there's questions. I tried to bold them to make it clear. And at those sections, stop, look at the raw data, see what it's saying, and try to make your interpretations. If you have any trouble, I'm going to be filtering through both on Zoom and in the classroom. Excellent. Yes, so as whoever's on Zoom, I hope that you've been able to get down to the SAM-BAM piece of the file format. That'll be helpful for the homework and going back. If you have any questions, feel free to email me, as always. And just before I turn it over to Karolina, I'm going to go through just the pieces of the homework or the 3 problems that you'll get formally tomorrow night, just written out, but I've had the time to put them on the board here so you can start to work on them and think of them. So it'll be to make your own Google Colab notebook. You have the test data in the C2 folder to work with, and you'll basically, for homework 2, be developing some of the tools that you would need in order to start doing some raw signal analysis. And then in the subsequent weeks, we'll give you more interesting data sets. If you downloaded the PNG file that was generated by Wally being a corresponding command line version of the integrated genomics viewer, you would see that the reads in question are not the most exciting. They are, there's a lot of errors, there's some, and it doesn't cover the entire virus transcriptome. Those are really just some raw reads from 500 randomly selected reads from the file. So this will be a way to generate, data sets with more interesting signals. So the first question is, as you showed in the last question, you subsetted the SAM file or the BAM file to specifically capture all of the reads that align to the reference transcriptome. And as you probably learned through the notebook, when you align to the reference transcriptome, the SAM file actually keeps all of the reads that were valid and that were input. So it includes all of the reads that were base called by Dorado, and it doesn't necessarily remove any unaligned reads. So as a result, you need to use the command that you wrote in order to isolate all of the specific rows that had reads that aligned to HCV. You can use those read IDs. And then a little hint here is to go back to SLOW5 tools, look through that documentation, and find the command that specifically allows you to isolate reads from BLOW5 file by read ID. And so basically to write a function, to write a script that will create a BLOW5 file that only contains signals in it that actually are associated with alignments to the HCV reference transcriptome. Then in question 2, you're going to use the formula. So there's a SLOW5 file specification, which you can also find in the notebook that's linked to. And in there, if you read through it, you'll find a formula as suggested by the bonus, which allows you to convert the raw signal to PA. All of the information that you need in order to convert to picoamps is contained within the SLOW5 BLOW5 file, and the formula is contained in that documentation. So use that and write a script that will convert the raw signal to picoamps. And then once you have now these specific signals in picoamps, think about different ways, and there's many different approaches to doing this for how you might create a dimensionality reduction mapping of these raw signals into 2D space. I should probably specify that it's a 2D dimensionality reduction. There's a few different ways to do it, but document your assumptions and describe the clustering patterns of these specific raw read signals. Karolina,
Yes.
take it away.
Of course. All right. So the second part is going to be in the separate Google Colab. I looked at the intro tools that like the questionnaires with our experience. It seems like it is a bit varied. There are some people that are more wet lab. There are some people that probably like prefer coding in Python because they're coming from like heavy computational background. I used to be a Python programmer and I hated R. And then I learned about tidyverse, and now I only plot in tidyverse. So, the purpose of this part is basically to give everybody awareness of how to quickly extract what you need from your data using tidyverse and some like basics of plotting. So I assume like can I ask from people that are both on Zoom, if you can put it in chat and in here, did you ever created a plot with ggplots? So, all right. How many of you use tidyverse? Some of you. Okay, half here. That's okay. Right. So yeah, so this is going to be quick. To go to this link, you should be able to see the Google Drive. I'm going to put this in the chat in the Zoom. That's probably too much for 45 minutes, but I will post the filled notebook after the class, and I will ask Theo to like send an link to it as well. All right, so let's switch screen. So this is what you should be able to see, and this is the notebook that we're going to be using. And there's a basic data set that we're gonna use. It's a bit big, so like go ahead and start it and try to like install the libraries first. And on purpose, there are empty places for people that are actually very familiar with tidyverse, and you can just speed through. At the bottom of this notebook, there is, and I'm going to be talking a lot. And at the end of this notebook, there is a very short homework for this part. And this sort of basically trying to check parsing and extracting data from VCF that compares the variants supported by different sequencing platforms. And then trying to figure out what is the best way to visualize certain thing. And I welcome your creativity in terms of visualization. Right, so the setup to this is very similar to what you had. So I'm actually running, even though this is in R, I'm actually running this notebook in like default Python, just so nobody is confused. And I'm using Bash at the beginning. I mostly do it because I want to mount the drive using Python, because mounting the drive with R is just pain. It's possible, but it's just much more complicated. You need to install additional side packages. So that means that I need to load the magic, and I need to use this R decorator at the beginning of each cell to basically convert all comments in cell into R. So once you get to this part and you're able to like do an easy plot, that means your R is working. And here are the packages. One of them is kind of like a clue. So we'll see. If you see warnings as they are here, this is not a problem. So tidyverse is a set of packages. And as I attach them here, you can see that we have specific versions, and it always shows the conflicts between the packages. So you're ever aware if the functions are named the same way. It's not a problem for us in here. Sometimes you have to like specify, I don't know, like a function select is just so common. Like sometimes you just need to specify which library you're using it from, which in R you do by typing the name of the library 2 times a colon and then the name of function. Here everything should be fine. So why use tidyverse? So basically tidyverse, if you are coming from computational backgrounds and you are familiar with like databases or with like bash scripting, this is how you use tidyverse. Tidyverse is the concept of like pipes, basically, like you would use in bash, and it has just a couple of basic operations that when you learn them, you basically can do whatever you want with your data, and they are very similar to like database like operations. And what is very nice is that, and we'll go through, I think, the most important operations in my opinion, and look at them in a moment. And what is very nice is that one of the packages that are part of tidyverse is ggplot. So there's like, you can just pipe it right through to the ggplot function and like continue. So it's very cool. A couple of, okay, so yeah, so this is basically how it works. So here I'm just creating a sequence of 1 to 100, calculating the average of all of the numbers, right? Pick it up and then parsing as string. And this is how it would be in normal R, and this is how it works in the tidyverse. So we just pass it on, and each time you use the pipe operator, basically what is on the left comes as a first argument of what is on the right. Basic, and I think we're gonna be fine. All right. So so basic operations. This is a bit smaller, but I think it should be mostly visible. So it truly is like 8 operations, and you can do everything. So first thing that you would like to do with your data is select specific columns, or maybe drop some columns. And for this, you would use select, and it would be select and the name of the column, or minus name of the column for dropping. The option to selecting specific rows is filter, and then you can put a condition that has a logical output. to drop. We will see it in practice in a moment. And this is how you select, let's say you filter the rows for only the variants that are chromosome 1, for example. Then we have ordering data. It's useful when you're just, looking into what you have. Usually it's not very much useful in actual coding that much, because you would like sort or like parse it differently. But here you would just arrange it for viewing so that you can have the highest, for example, values to be at the beginning or the lowest. Next, now that you know how to extract a part of your data, you want to change it. So adding a new column or changing a column that exists is done using a mutate function. And separating a column in multiple columns. So let's say we have, which we'll see in the moment in VCF files, columns that have values that are separated by commas or separated by it's columns, right? I always have a problem with 2 dots, in English. It's just so irrational on my part, I guess. All right. And so you would like to separate one column in multiple columns. You just separate. And similarly, there are additional arguments there that we'll see, additional options that would like drop some of the columns, merge some of the columns as you do it. So it's very handy. Unite is the exact opposite. Next, a very important concept is grouping. So this is where the databases are coming from, right? So let's say I try to use a function that is called summarize to calculate how many rows I have in my data set. If I just run summarize on my data set, I will just get a total number of rows. But then I can add a grouping variable. So let's say the chromosome column, and then I will get the number of rows per each chromosome, right? Very easy. And the last part is joining. And this truly is, again, like a database. So the concept of joining is, let's say you have one data frame if you use R before, and here we have table, and you have the other one, and they have similar columns, but they might be like different sizes. And then you connect one to the other. If you connect on the left side, you will have all of the columns that were on the left in the first data set, and the matching columns from the right. The right is the exact opposite. And then inner and outer would be either having the columns that do not have matching rows or only the matching rows. All right, and I have a bunch of links in all of this. That's truly is only so that you know that they are resources that you can use. So there's a bunch of links throughout this notebook. And this particular one here is just a workshop that I ran before. It has basically what we will go through here with more like simple examples for people that will get maybe a bit lost if we go too quickly, but we only have 45 minutes. Right. So this is the checkpoint. How is if everybody has libraries right now? Everybody has libraries. Are we good? Okay, great. So if you went through the first part, or you will finish it in the future, you probably ended up at the SUM-BUM level. So this is the aligned reads level. After having an aligned reads, usually you would align them to the reference genome. Sometimes you can align them de novo. Since we are talking about very long reads, like Chris mentioned last time, how you would use de novo alignment and long reads would be probably sequencing the same genome with short reads and long reads, and then creating like a context with long reads that you then polish with short reads to create new genome assemblies. So that's the side note on the BUM side. But usually, in most cases that you will probably work on, you will just align them to the reference genome and then proceed to try to see what are the differences of our sample versus the reference genome, right? So we will call variants in this case. So I'm gonna go the different route and try to make you talk to me. So what type of variants do we have? What type of variants can we expect to see in the VCF file? You can type if you're on, or speak if you're on Zoom as well. But yeah, SNPs, yes. So single nucleotide polymorphism. Do you know what is the difference between SNP and SNV? Single nucleotide population.
Population frequency.
Yeah, population frequency, basically, yes. But some people kind of use it interchangeably. They shouldn't, but it kind of is a very similar concept, right? It just, so the way I'm thinking about it is coming from classic genomics. So it's just like change of one nucleotide into other, right? So that's what we are talking about. What are other non-SNV type of variants? Indels, yeah. So what are indels?
Small stress or elevations.
We have to be louder because I can't hear.
Usually less than 100 of the
Yeah, well, so this is the size, but what is down region?
Western.
Yeah, so this is the size of the variant, what you
Yeah.
said, yeah. And what is the change? Why is it a variant? What is it called a variant?
Because they just change.
All right. Yeah, so I'm asking what is an indel?
What is an indel? There's so many. Yeah, it's insertion or deletion.
It's you start talking loud and then
Insertion or deletion, right? Like,
yeah. So there can be a deletion, there can also be an insertion, right? That's why it's an indel, in and del.
Yeah.
So if you compare it with the reference genome, if your alternative, like in your sample, is longer, then this is the insertion, right? And if it's shorter, then this is a deletion. All right, just wanted to establish this. And then this is usually what you would expect from the VCF. And depending on the software that you use to call the variants, SWABA comes to mind as an example. Usually you would either get two separate files for indels and for SNVs, or you would like to specify like a mode that you want to use. In single molecule sequencing, the concept of variant calling is very interesting, and I don't think it's that much like widespread research, but we can like figure out how it would be appropriate for this class. So here we're going to use like non-single molecule sequencing, right?
why is it less studied?
Yeah, so because I would say that a lot of people are interested in like single transcript level, and if you call variants, you would generally, like by default, you look at like a population, right? Like all of the reads that are at the position. But what I wanted to say here is that the single molecule sequencing gives us a good advantage in the phasing stage. So if we were trying to like figure out which alleles actually are coming from, which variants actually are coming from which allele, if you have additional UMIs, whether this from single molecule or transcript level, or you do error correction sequencing, right? This is always an advantage. There's also a consideration of if we are looking at base modifications, right? Like what is the impact of a base modification on DNA damage, right? That DNA damage is a leading cause of variants, other than, you know, usually the variant appears when there's damage and then there's a repair, especially the insertions and the deletions are treated mostly like this. So, and here I'm talking about somatic variants, right? Yeah, so especially if you can also like include additional barcodes, right? This would be very interesting. And when I wrote it before class, Theo mentioned the sprites, right? So do you wanna?
Oh, so I mean, we'll talk a little bit about it next week. So that's why we actually didn't go into the discussion of like what the quality scores mean, because the quality scores themselves have a much longer like history than, and that is kind of appropriate for a lecture setting. But for a long time with the nanopore systems, you were getting T-red scores on average between 10 and 20.
Yeah.
And so at those sort of score value levels, the amount of coverage that you need over a particular region to confidently call a specific single nucleotide polymorphism is quite large. And you know, especially if it's an allele in a population that's only perhaps represented half of the time, there aren't. Yeah, you either need like typically very high coverage, which is going to be maybe more than what you could get on your average min-ion, although you could get it on your bigger Prometheus. Or if you want to do something like understand insertions and deletions, that's actually very difficult to tell, because sometimes, you know, the way that the electrical currents and the signals are run, you just get insertions and deletions that happen just by random chance or frequency. Because when you look at the actual nanopore, it's 5 base pairs, so it's k-modes of 5 that are being read simultaneously. And so especially with homopolymers, so long chains of the same base pair, the accuracy, although I think all the systems kind of have that error associated with it, it's very much accentuated in nanopore. You won't get an accurate estimate for the size of the homopolymer.
So the change of the base pair is like, on the basis of it, is a change in like physical conformation, right? So like just a consideration of how physical conformation impacts the like accessibility to DNA repair mechanisms, for example, right? Is like a very interesting question.
The natural population too, you're going to get these base pairs, these like transcriptomic and DNA modifications as mentioned, that are going to go through the pore and change the signal in a way that the model might not have accounted for necessarily. So if you have, for example, an M6A methylated base or an 8-oxy damaged base, and it goes through the nanopore, it might look like a C, it might look like a U, it might look like some other base pair. And so some of the earliest base modification algorithms to detect those base pairs looked at errors specifically and looked at errors in the context in the context of calling the base pair modification. So that if you saw a SNP, it must be because the base pair is modified, not because the base pair actually there actually exists a SNP at that position. So yeah, but it really depends on, at the end of the day, and that's why this is a longer story for another lecture, of how that base calling algorithm works, what the raw data is that it's trained on, and then understanding its generalizability.
But at some point, you will hopefully arrive at this level. So you will have the VCF file, right? And this is the part that you that we basically go over it and try to figure out what exactly is here. So yeah, and I know that I've seen a couple of like non-standard VCF files, like some of the newer technologies, like Ultima, like to give us new surprises, giving us non-standard files. But generally, like this VCF file has this specification that is pretty well established. It's 4.2. If you go to this and open it, then you can see that there is it's a very long document, and it describes most of the options that should be here. What is important is that in the like info and format, there could be like non-standard fields. So this is just like additional values that are added here, described right in format and info, and then usually this information about what they mean will appear in the header, right? So this is how you read it. So side note about tidyverse, it's very handy to because it's very readable and it's very good in this way to like explore your data. What is not good about is performance in terms of like time. If you have very large data sets, it takes a bit longer to parse it. So in this case, I'm using data table, which is basically like the alternative. It has most of the comments that we have, just a different grammar to just read this file. in my opinion, data table is, there are kind of two camps. So I'm in tidyverse camp. The data table is a bit like the readability is yeah, it's harsh. So I like to use the tidyverse. But the performance in terms of like reading the data specifically, it's completely not comparable. So here we use Fred to read the data. So that's information. Oh, and one more thing about the about R for people that are that said that they are not familiar is that normally in programming languages you would have, if you specify a variable, you would have the name of variable on the left side, and then we would have equal signs or something like that, and the what do you want to assign, like we have here in the f name argument. In R we also have this arrow that can work in two ways. So you can have an arrow used here in the f name line for with arrow going to the left, and then you can also use it as arrow going to the right, and then it will assign to the name of variable on the right. All right. Okay, let's go through this quickly, and let's do some warm up because you're falling asleep. So what do we do? So we have our data now here, and we can check what are our columns. I like to, so you can notice that this chromosome has this additional well at the beginning, like octocorp, that's how I learned it's sometimes called, yeah, hashtag. So I remove it so that it's optional, but it's easier to, you don't need to use quotation marks to refer to the column later on. Yeah, so this is how our data looks like. So we can see that table, a basic data structure of tidyverse, is very similar to frame or like an array in Python. What you have here is the additional information about like types. There is a different type it is displayed, but and again, it won't display a whole data by default, which is very handy for just exploration. What is like a tip from me is that if you have long columns, they might like show fully on your screen. So just deselecting columns like this, using pipeline in this case, it won't modify your data. So this is just for like temporary operation. So here I remove some of the long columns, and I can see more of the format column, for example. All right, so let's go through the warm-up. So you have a link to the specifications of the VCF, and you loaded a file. Can you tell me how many samples and how many variants are present in the file? No?
I couldn't load the file.
You cannot load the file?
Yeah, first of all, I can't set the working directory. That was the main problem. I don't know, you have the same problem?
No.
And no such file or directory.
Oh yeah, that's the problem.
I can set the working directory, and then the file does not exist or it's non-readable.
All right, let me check. Directory should be readable for everyone. Let me check. Is this the same issue that was before?
Not sure. Now I have to change. Yeah, cannot change working directory, and then I try to like paste it to read a file. It says non-readable or does not exist.
All right, can you try now?
Is she? Cannot change working directory.
All right. So one of the way you could try to do it is I gave you a link to a whole like Google Drive directory. You can just clone it to your Google Drive and try to run it from your side. And it contains the Google Colab. Yeah, so how many variants are in the file?
504, 941.
Yeah, it's also on the screen. Yeah, how many samples? 5? No. So compare it with the default screenshot or like the specification that was shown before, right? So our static columns are here like up to, so we have the chromosome, we have a position, we have what reference was changed to which alternative allele, quality, their filter, all of them are passing in our file. And then we have our info and formats, and everything later is simple.
Okay, I mean, there's 3. Sorry, I think there was something wrong with my code.
All those three.
I selected from the 8th column to the end column. I thought, because I thought maybe there was hidden columns.
Yeah, so there are three columns here. So this is the genome in a bottle Ashkenazim trio. So HG02 is a son, and HG03 and 4 are parents. So that's what we are going to be looking at today. There's more information linked at the bottom in the homework section. So, okay, so we know how many variants, we know how many samples. There's one more question, I think, there. Oh yeah, how many chromosomes are present in the data sets? How do we check this? Nobody wants to.
We said unique.
Yeah, unique and then length.
All right, so the way I would do it is I would just look at the chromosome column. Again, there are different options. We can do it fully like for tidyverse. Here, I just use a pipeline from tidyverse for the normal operation that I would do in R, which is I will just check for the unique chromosomes that we have. If I am able to get them. This is the struggle. Oh, I did not run everything. Correct. I did not run everything.
I got.
No, like you put length and I think.
Oh,
you
yeah.
can't just,
You
you can't
check,
pipe it, you just pipe unique, it will give you.
Yeah, you can put length here, but then you will just get the number of rows, right? And what I'm trying to get is, so you can also get a table to work here, another standard comment from R, and you can get how many of each chromosome we have here. That we'll try to see how do it in tidyverse way to just wrangle. All right, so knowing this, I would like to ask you which genotype fields are present in our file. So we can take a look at our format here. So first of all, we know that all of our format fields are the same here, and they have this particular field. So if you open the specification, you should be able to find most of them. Okay, this is gonna be rapid fire. So what does all of this fields mean? You can open the specification and you can look at it. So what does GT mean? That's the most important one. Genotype. So what types of genotypes we have here? In the file, if you look at the VCF specification, you will see that there are different ways of notation here. The most important was the concept of phased versus unphased. So like we mentioned before, that did you know which allele it's coming from? In most cases, you will be working with unphased datasets like here. The difference is the either you have a slash or you have like a pipe symbol. I don't know if you're on Zoom, you should be able to still see the board, right? Correct? Okay, so if you have like haploid genome, right? Then you would just have like one number. if you have a diploid genome, you would usually have two, right? And then even if you have datasets for human, you sometimes can have biallelic sites, and you can have like triploidy, etc. locally. So then you'll still be able to like see how many numbers are here, right? So that's in terms of haploid or diploid. The unphased is slash. So for example, this one, and the difference between this one and this one is that this is the phased genotype, right? The 0 here is the reference, and 1 is the alternative allele number 1. If we have more than one after commas, then in alt field, then there's going to be alternative allele number 2. And if you look at the AD field, this is the allelic depth. You also have the same number of alternative alleles there, comma delimited. Right, what is important here? Oh yeah, so a very important question. So if we take a look at different types that we have here, what do we need to do? So since we know that this is the first part of the string, then we need to separate and pull out all of the genotypes to see what are our options. Right. So this is in the basic exercises part. So here's to separate, and what I recommend you to do is to run the code like this, one comment by one comment. So you would remove this part and you would run it first, right? To see how our data looks like after the separate. Then you would add another comment and run it again, another and run it again, etc. So in this particular case, it will stop working because I interrupted it. That's okay. All right, there we go. So in this particular case, don't worry about it. I stopped it. We have genotypes here. So if we pull them, you will see that some of them look like this. What does it mean? We have a dot, slash, and a dot. Any ideas?
Not in the.
Yeah, it's not in the sample. So
Sorry.
it's not in the sample. Yeah. Your intuition is good here. Yeah, so since we have multiple samples in the same file, then we basically have a row per variant, regardless of in how many samples it is. So dot slash dot here for genotype means it's not in this particular sample, but it is in additional one. So here you can see that in sample number 3 there is this variant, and it's not present in sample number 2 and sample number 4 was somewhere there as well.
Oh, they don't do 0 to /0 here?
Yeah, so because 0 refers to reference allele.
I see. Oh, I see.
Yeah, so this is basically an equivalent of NA. But if you have NA, then when you parse this file, if you did not put it there in purpose, then there's an error. Yeah. All right. Yeah. So this is how we work with tidyverse. I would love to spend more time on this, but
Sorry, so on the board when you did 0/1/2, what's the 2?
so if you have two, like if you have triploidy, right? So you would have allele 1, 2, 3, right? So
Yeah.
0 refers to reference, 1 refers to alternative allele number 1, and 2 refers to alternative allele number 2. And the number of them, and you can still have 2 here, right? If you have, let's say you have a in genome where you have in some reads you have A, in some reads you have C, right? Like a somatic mosaicism, right? Like a somatic. So then you would still have 2 numbers, but it would be either 1 or 2. In this particular case, if you have 0/1 or 1/0, that is the same.
Because that's not phased, right?
Yes, exactly. And 1/1 is the alternative. 0/0 is homozygous reference. 0/1 could become 1/0 if you change the reference, right? Like, sorry, 1/1 could become 0/0. That's what I wanted to say. So because it's a homozygous, right? So depending on the reference, it's either alternative or it's a reference. Right. Okay. Not enough time to go through everything. So let's just go overview wide. So at the beginning of this file, and then you can go through this file on your own pace. I will post the solutions. So we have some basic exercises here. So we're checking genotypes as we just discussed. Now, since we know that the sample number 3 and sample number 4 are parents, we can check how many variants are non-overlapping with parents. Something that I would caution you for in practice would be that sometimes, depending on the color, you will have like either like unique row per position, and sometimes you would have unique row per like option variant per position. So here we just have per position, so it's fine. But sometimes when you count variants, it's very important to specify what you count as a unique variant, right? All right, and yeah, and here we can choose to count how many SNPs we have and how many indels. And for this particular set, I have it in an easy way for you. So this is the equivalent of what we did with the table, but we check if we have SNVs. So there is no difference in length between reference and alternative, or there's a difference. And yeah, and then we have some more complicated exercises. So comparing across different samples. I think I will just show you how to do it since we don't have time. Here's what I would recommend. All right. So how to read this? This is very important. This is a concept of wide versus long data. If you understand this, you will be able to basically do whatever you want. And this is important for homework as well. I don't think you're able to do your homework without it. Or maybe you can, I challenge you to do it without it. So the concept of a wide versus long data. So if you have wide data, this is basically the type of data that we have here. We have many columns, and then we have here like one column per sample, right? So to turn it into long data, what we want to do is we want to have a column. We'll gather all of our data from these 3 columns, the HG02, 03, 04. So we turn it into like the column that has a variable of which sample it is. It will add more rows, right? Technically, it should have 3 times as many rows if we have three samples that we want to have here, and we have all of the rows filled. So this is done with gather and the spread commands in tidyverse. And if you figure out how to use them, then you are basically good to go. So gather would take the columns that you have here and change it from the wide to the long. And then spread, you do the exact opposite. Usually you specify for these 2 columns, comments, you specify key and value. And there are examples here in the notebook. Yes. So here in this particular case, I'm specifying that I'm calling my 2 new columns, sample and sample data. In sample, I will have the name of these columns that I specified here. And then in sample data, I will have the values from these columns. And in this very easy way, I have suddenly 3 times the amount of rows that I had before. Why would I want to do that? If anybody is familiar with ggplot later, you will know that basically I'm creating a new variable for each row. And then I can just specify, oh, put on axis X this particular variable, and on axis Y, the other thing, right? But also like for parsing, it's very helpful. So there are simpler examples in the workshop that is linked for this that I pointed out before. If somebody has any problems, you should take a look there. There's also a bunch of materials at the bottom linked for ggplot and for tidyverse. So, and of course, you can just email me and I will try to help you. So last couple of minutes, since we started a bit later, I'm just going to do maybe 5 minutes about good plotting, and you can fill the exercises by yourself, follow through. So the way we create plots with ggplot is layered. So we first establish basically what data we are using, establish which variables we want to use, what type of plot we want to do, which is very important for the good visualization, and then we can add additional for example, change font, change color, like additional embellishments. And here you have a very easy step-by-step workshop that I can go through. I think I'm gonna switch to slides and I will just, yes. So this is basically the same plot, just added again and again. And I think what is important to establish here is that there's a couple of resources, right? So there's some packages that are very cool if you're preparing figures for journal. You can do a whole figure just in R with this. And there are some like color palettes that are colorblind friendly and also like specific to journals as well. And I'm linking also to books. Again, you don't need to read them, but if you need to like find more about how to do good plots, this is where I would start. The most important part about plotting is that we need to figure out what exactly is our purpose with making a good plot. So here I put, I think, one of the most major visualization plots in history at all. So this is the 1854 Broad Street cholera outbreak, which is basically an outbreak that this is a basis for modern epidemiology. So Joe Snow went around like ask people and like mapped on map where they lived and basically find out which of the wells in the city were contaminated in this way. And if you can see, like, this is a very good visualization because you can see, you know, at the dots of where people lived and try to like triangulate all of the like wells that are in the area. Obviously, you probably won't do something similar, but this is like a very good example of like very easy visualization. What I wanted to show here from the modern one is this one in particular, which is the difference in the life expectancy in US and like other countries. This is a couple of countries together, mostly European. And the way we could plot it could be just average like life expectancy of one to just create a line of the other, right? And then the difference wouldn't be that big for both of them. This plot, I saw an updated version. Right now, it just is actually like up to like -2 here. So you can see just by the decision of focusing on the difference, that's exactly what we want to show. And we plot different variables on the y-axis. It is strikingly visible from the beginning, if you look at it, that up to a certain point here, like to more or less year 2000, the life expectancy in the US was better. And then there was a change and suddenly, and it's trending downwards, right? So, so this is a very good example of what exactly does it mean to like think about what you are plotting and how to do like a clear plot. And the other variable other than like purpose would be the type of data that you have. So, oh, why is it so dark? So the type of data would probably be the type of variable that you're plotting, like categorical variable, for example, sample, continuous variable, number of mutations, the number of variants. You can select, there are some kind of plots that are more appropriate for different types of plots. Something that you have in homework is basically selecting a plot of your choice for the problem, and basically writing a short explanation of why you selected this particular plot. This is because the problem that you have specified in the homework is something that normally, so here would be solved with like a Venn diagram, but you have more than 3 groups, so it becomes like much less readable. So there's actually a hint in this workshop of what I would do, but I removed it, so maybe you will find it. Yeah, so basically what I'm asking you to do is after you go through this workshop on your own, take this data and try to extract the information about how many platforms support each variant. This is not a field that you would have in your standard VCF file, but since this is a VCF file that is coming from consortium and it's very well established, you have such information in like info column. And yeah, there's like one question about long-read sequencing technology, but what I'm most interested in is how you would decide to plot this particular problem, right? So like we are trying to compare how many variants are supported by how many platforms, and what can we do? And this is the short homework from this part, and I will ask Theo to add it to the email. All right, I know I'm going over the time. I will just do a last thought regarding VCFs. Usually, when you get your VCF, you will have different ways to evaluate it. One of them would be just taking a look at the filter field, but this is like very basic. So filter field, just the thresholds are very low, right? So most things will pass. So just by default, I usually just drop things that are not passing. If there is so many of them, then I would take a look. Second thing that you can take a look at. So here we split the fields for different parts of genotype field. You can see that there are different ways that the quality and the depth is described. So here we are trying to take a look at the allelic depth, mostly because we want to calculate variant allele frequency. So which is a metric that allows us to figure out the proportion of variant alleles within the like loci. If you have biallelic sites, you probably would like to take into account not only what you have here, but like all alternative alleles. So you would you basically take the allele depth and divide it by a sum of reference and all different alleles. In this particular case, we just look at the major. And yeah, and there's something very suspicious here that I saw the first time I looked at this data. And this is not just this data, this is by design. It's not a bug, it's a feature. And it's that the DP field, it's a depth, and AD field is also a depth, right? So why does the AD fields do not sum to the total that is in the DP? Anybody knows? probably not. So I will just give a response, and but this is something that I will link to you to if you are interested to take a additional look at. So basically, what it means is that for AD, allelic depth, the only reads that are counted for it are informative, and a read can pass through the filter check but still be uninformative, and it's all about the likelihood of alleles given the read, the likelihood of allele actually being a variant, and the difference. If you calculated the difference between like first most likely to the next most likely has to be like 60%. Like, so the first one has to be 60% more likely than the other one. That's when it's informative variant. So if the reads are for variants that are informative, that's when they're going to be counted to AD. That's why AD usually sums to like less. And that's why the AD is the measurement that should be used for variant allele frequency calculation. Yes, and I have this written down in the full version that you will get, so don't worry, but that's a very important point. Yeah, and that's.
Yeah, no, so I was just gonna ask, is the number of alleles that are the reference allele?
So the first one would
the
be
total, or?
first one would be reference, and the other would be alternative.
Right, but do we keep all of those for the allelic depth column?
So you would use AD, because if you look at the DP has both of those, the sum plus the uninformative ones. So that's like a total, basically.
Right, I guess what I was asking is if some of the reference calls are uninformative.
So, so it depends how you look at it, how much you trust your reference, but generally, like the like if it's a reference, like if you're completely different than a reference because you're a homozygote like alternative, then they are all kind of be uninformative, right? Or like if you have like completely different situation, we have super like somatic mosaicism, like bunch of different variants that you're getting in your data, you will probably get like a variant that is not that valid unless you like specifically filter for like low allele frequency, and like really like somatic variants are low frequency are very hard to work with. So so yeah, I guess. But like AD is what you would use for calculation. And if you would have more than one, there is a concept of MAF, like minor allele frequency, right? So that would be the second allele that I mentioned that you would add to your variant allele frequency calculation. Yeah, and that's everything. Sorry for going over time.